So today I would like to discuss in this part 2 the recent methodology that we have developed to better understand trafficking inside the endoplasmic reticulum, and that we are going to reveal here by the data modeling of many trajectories of particles moving inside the ER. So first of all, I would like to remind you that inside eukaryotic cells, the ER are really spreading the entire cytoplasm of a cell, and it is basically composed of two parts. One, which is called the rough endoplasmic reticulum, which is here shown on the right part, which connects to the smooth endoplasmic reticulum, which can be seen as junctions that are connected, nodes that are connected by tubules. And so it remains today very difficult to analyze what is the motion, how proteins, molecules, ions are transported across this network. So to answer this question in part 1, we have already discussed that we can use many trajectories. So let me remind you that we have inside, for example, the network here, we had these tubules, we follow trajectories. And when we followed these trajectories, what we have seen is that looking at these pictures here, we see the trajectories, the trace of the trajectories, which are colored according to the velocity, to the displacement. That is, we take each point, for example, x of t, calculate the difference xt plus delta t minus xt divided by delta t, which gives us the instantaneous velocity, and we colored according to the amplitude. And what we have found is that you can see clearly here two types of colored, indicating that there are two types, two regions. One in blue, which are the region of high density, which are characterized by a slow velocity. And then we can see the red color basically between specifically this region of high density. So the red color here are characterizing jump velocity, that is very high velocity, and as we can see here, they are mostly distributed between nodes. So at this stage, we can conclude that the ER, the topology of the ER, is segregated into two parts. One, the tubule characterized by high velocity, and second, the node, which is intersection of the tubules or the sheets that are characterized by a low velocity. More specifically, we decided to look at this region of low velocity, and we used an algorithm called dbScan, which is a very classical algorithm, to identify and to obtain a segmentation of these regions of high density. And then we use a different algorithm to fit these ellipses in order to determine the border of these regions. And this is important if we want to study the statistics of jumps between this region of high densities. Then we looked specifically on this region and estimated if there was a vector field, that is, if the reason why trajectories are confined is because there could be a potential force. So here what we calculated a force by estimating again the difference of velocity between time t and time t plus delta t. And if we divide this by delta t, and we average. So this is supposed trajectory k, the k trajectory, and we average this over k. This quantity behave or converges using the classical representation or model, stochastic model of the Langevin equation, converges to a force. So we can do that at each point here, at this point, at that point, and so on. And what we found, as shown here, is that we could see arrows of the arrows of different color that converge here toward the center that I'm showing now. And this is not only one isolated case. We could see it repetitively on the different position, and actually on most region of high density are characterized by this converging arrow. And these converging arrows, as we can see here, these converging arrows is actually the signature of a structure that is well known in chemistry, which is called a potential well, from which we can calculate a force by using the gradient of this quantity. And this gradient of this quantity is the force, a virtual force, the origin of which is unclear, which will prevent a trajectory to escape and bring it back to the center until the trajectory can eventually leave the domain. So this is a very strong characterization of this region of high density. And so to confirm or to have another possibility to explain this region of high confinement, we use what is called the classical MSD analysis, which is basically we calculated the difference between t and t plus delta t over the trajectories. So you can calculate this difference squared. So you make it squared. This is the MSD, and you average over time, over realization, if you have enough realization. And this quantity, it is known to behave like t to power alpha with a coefficient in front. And when alpha is 1, this is understood that the typical realization is Brownian motion. When it's lower than 1, it is called subdiffusion. And indeed, this is alpha equals 1, and we found here that most of the slope of the exponent alpha were below 1, as can be seen here and also here in the second graph, as you can see here. And we reported here examples of the anomalous exponent alpha, which is really between 0.3, 0.4, 0.5, and this was obtained for different cells, Hex cells, COS cell, HSS cells with this calreticulin or allotag for the ER that confirm that all molecules are subdiffusing in this region. And the reason of this subdiffusion is this potential well characterized by these converging arrows. So in conclusion, this subdiffusion characterizes the motion of inside the well and is correlated with this confined diffusion. The fact that the Brownian trajectories is confined inside this region of high density or nodes. I would like to say something that we don't understand the reason of this confinement. Now, the second step was to study the what happened between this region of high confinement. So we started here with this region A, which is a node. So this is the starting point, and we looked at all trajectories starting from here and looking at what will be the next step. So basically the trajectory starts inside A, and here this is one example. One trajectory went to the right, another went up, another one went to the left, and we could follow their paths as a function of time. And associated to this, what was quite interesting is that, so if we look in here, we saw that the moment of escape was associated with a very high velocity. After the velocity drops, and then there was this region that we characterize as region in between the nodes. That is, this region here are characterized by being here in between nodes, where the velocity was dropping. So again, at the beginning, when the particle escape, that is in this region, the velocity was very high. And so those are three examples, 1, 2, and 3, where you can see that condition on a trajectory starting specifically here at the red, where the red ellipse is marked, we could follow how trajectories are actually going into these different directions. And they are characterized by an anomalous exponent which is larger than 1, which is characterized as a super-diffusion in basically all possible directions. Next, we wanted to study how the velocity evolve as a function of time along trajectories. So we started here, we looked at trajectory that are initially starting inside the red ellipse. This is in these two cases. And we marked the different time steps using the numerotation 1, 2, 3, and so forth. So we could see after one step, two steps, how far away are the different trajectory from one single node. And as can be seen here, the trajectories after some time, they are found in neighboring nodes. Here this is an example where it is quite isotropic. Here the anisotropy is not very clear because probably there was not necessarily some network here. But what we found in general is that there was a clear isotropic distribution, that is, trajectory could escape in all possible directions starting from a single node. Finally, we looked at the following property. If we isolate two nodes, A and B, and ask what are the trajectories that go from A to B and the trajectory that go from B to A. So, is there a moment of time where all trajectories going from A to B move or are synchronized, that is, they go always from A to B? So can we identify this moment of time where all trajectory goes from A to B and then trajectory go from B to A? And how long does this period of time last? So what we found using this analysis, we found that there is a distribution of unidirectional displacement. And this time scale was about 3 seconds. So the interpretation of this very long time is that inside these trajectories, we are not necessarily seeing all of the molecules because not all of them are activated. So we believe that actually the time, the unidirectional time, should be between, let's say, 30 and most likely 300 milliseconds. That is, for this amount of time, trajectory goes from one direction, and after this time, they switch in the other direction, and probably with a Poissonian rate. Associated to the switching, this here, we looked at the analysis, we looked at the velocity, and the analysis revealed that the motion between nodes was much higher here than the velocity, the amplitude of the velocity inside the node. And here we looked at this velocity, the velocity peak, which for the velocity, the high velocity was about, it lasted about 35-40 milliseconds, while the period of low velocity were about 100 to 120 milliseconds. So again, I would like to come back to this notion of unidirectional flow. This is the mechanism of which, so if we start, let's say, this is A and this is node AB, it is quite unclear what is the mechanism that would allow trajectories to go only from, let's say, as we can see here, to go only from A to B, and then here, those are going from B to A. So, however, again, these features could be found in most of the nodes that we looked at, suggesting that there is a mechanism by which the flow is temporarily unidirectional before switching to the other direction. And we have proposed that there will be some type of peristalsis contraction where the ER could be contracted. So if we imagine that we have two nodes A and B, and we have a tubule here, that there will be here some contraction due to the actin network, so leading to here a contraction that will create a flow, and this flow can push the material inside the nodes, and that's why this could be unidirectional. And when this flow now starts to be squeezed, then it will create the flow in the other direction. So we'll have a flow here in this tubule. So to conclude, the luminal ER motion can be revealed by the single particle trajectories using a methodology where the basis is to have a stochastic model for the trajectories. So we could identify this germ diffusion process where we have diffusion inside the nodes, that the motion is restricted until it is going to be expelled and move inside the tubule. So here we have the tubules. It's going to, the motion is going to shoot to the next node very quickly. So while here we have this restricted diffusion, confined diffusion. And here we have this fast jump. So molecule stays at junction before ejection. The unidirectional motion, the mechanism of which, of the fact that it can alternate the flow, this remains completely unexplained to that day. So I'll refer for this second part to this following paper that we published in 2018, and this can be found in the following address. So I'll stop here, and we will finish by a third part where we will explain how from this single particle trajectory we can generate a graph in the sense of graph theory that can be used to extract more of the statistical property of the ER. Thanks.